Perfect. Yes, thank you.
All right, perfect. Thank you. Really excited to be here. As Sarah mentioned, my background is in epidemiology. I really specialized in disease modeling. Causal inference is something that is a really important aspect of epidemiology. It's something that I learned a lot in grad school. I never expected to carry it into my career as much as I have. And so for that reason, I'm excited to sort of introduce this topic and hopefully give you kind of a primer to causal inference as best as I can in about 10 minutes or so, and then introduce you to an application that I personally worked on at Genentech and Roche involving a single arm trial in cancer. And so in that application, I'll try to do it tutorial style, where I'll try to sort of introduce the method, why it was useful, and how we applied it. Okay, so first, causal inference. I'm also going to assume very limited knowledge of causal inference in the audience. So that's where we're going to start to get everybody sort of on the same page. So causal inference, I like to say that's an art and a science. So it's the ability to identify cause and effects, as opposed to the mere association between two events. So in other words, teasing out correlation from causation in this clever sort of cartoon. In these sentences, 100% of people who breathe die versus 100% of people who don't breathe die. So what do we mean by cause and effect? It's really the ability to say that action A causes outcome Y associated with A, action B causes outcome YB. So if I take this medicine and I get better, did the medicine help me? But causal inference is not actually asking if I do A, then some Y happens. It's actually asking if I didn't do action A, would YA still happen? In other words, what would have happened if I did something else? So these alternative outcomes are known as counterfactuals because they refer to what would have happened under different circumstances. So they say that the ideal way to actually do causal inference is with a time machine. So to turn back time and replicate the exact conditions under which each action was taken. Of course, we don't have a time machine, but we do want to know if medicines help sick people get better. So in the world of drug development, randomized controlled trials, or RCTs, are really considered the gold standard for establishing causality. So in an RCT, you start with some population of people with the disease, you randomize them into two groups, you treat one group with your experimental medicine, and then the other group with something else like the best existing treatment on the market, and we'll call that the control group. And then at the end, you can compare outcomes between the two groups, like response to treatment, survival rates, etc. So in this way, you can state that the outcomes in the treated group are caused by the treatment, and the control group actually represents that counterfactual outcome if those same people were treated with something else. So remember, causal inference is establishing what would have happened. The key piece here is randomization, which really acts as our surrogate time machine. So by randomly assigning subjects to treatment or control groups, randomization creates two groups that are similar in all respects except for the intervention being assessed. So in this way, we can actually isolate the treatment effect from the effect of other variables which might confound that effect. For example, if one group is older or sicker than the other group, then it's not a fair comparison. Those patients may die sooner, for example, irrespective of treatment. So randomization ensures that the distribution of factors like age, sex, those other clinical characteristics, and patient characteristics are similar between the two groups. And this is true for both measured and unmeasured factors. So in theory, even if you don't measure everything about the patient characteristics should be similarly distributed across the groups. But don't take my word for it. Can you guys see this, this video? I wrote some R code, which I'll put in the Slack channel, to actually simulate a group of 1,000 patients between the ages of 18 and 60 with some distribution of key clinical factors like age, sex, an oncology prognostic score called ECOG score, and an example comorbidity like having a heart condition. And so these are just an example of the types of characteristics you would want balanced in your clinical trial between those groups. And then I'll do a simple randomization of the patients into treatment and control groups, and you're seeing here of equal size, 500 each. So that's our sort of like simulated treatment and control group clinical trial. Okay, what I really want to show you is the distribution now of those 4 characteristics that I just simulated. So I just randomly assigned the 1,000 patients into 2 groups, and in these 4 plots, these are the 4 patient characteristics. You can see, comparing the left and the right panels of the control and the treatment group, that these characteristics are pretty evenly distributed. You have a good balance of males and females, heart condition, pretty uniform distribution of age and ECOG score. Of course, there's random variability, and they won't be perfect, but with a large enough sample size, especially considering that RCTs will enroll thousands of patients, this will be good enough. Okay, so we're done. Randomization is all we need for causal inference. Or what happens if randomization is infeasible or unethical or just simply not done? So my favorite gag paper on this is actually a paper that was really published. You can find this in the literature, reporting on an RCT on the use of parachutes, right? Randomize some people to jump out of planes with parachutes and others without parachutes. But of course, that would never make sense. We can't do that. But in a much more real sense, imagine a rare childhood disease without existing treatment options where it would be unethical to have a control group, perhaps. So this is where the field of causal inference is really prime and developed as a way to make causal inferences from observational or non-randomized data. So in the absence of randomization, how can we make causal inference? So causal inference as a field typically refers to this suite of statistical methods that enable us to do just that. So a couple of terms, methods you may come across in your pursuit or your studies would include propensity scores, G-estimation, instrumental variables. Those are very common in economics, diff and diff, which stands for difference and difference, and much more. So now that we've gotten our toes wet, I want to share with you a real example of causal inference in action in drug development. So to avoid over-commercializing this talk, I'm just going to refer to the medicine in question as M, which is a novel bispecific antibody that we developed for the treatment of lymphoma. So follicular lymphoma specifically is a very slow-growing but incurable form of non-Hodgkin's lymphoma. There's no standard of care among patients whose disease gets progressively worse. We'll call those patients relapsed refractory patients. And M was a medicine developed. It's a novel bispecific antibody that I have illustrated on the left. It's really neat. It actually simultaneously binds to the tumor cells and to your body's killer T cells to like bring them very close together and activate tumor cell killing. So it's a really nice mechanism. But what's important about this? It's sometimes the case that a drug can receive accelerated approval. So when a drug is shown to have a profound effect within a population of high unmet need, this was the case for our drug M, which was set to receive regulatory approval by FDA and EMA after its phase 1 and 2 studies, which are when you exclusively treat a handful, maybe 100 patients with disease, and there's no control group. So in drug development, RCTs are going to be happening later in phase 3. And bottom line, M was going to be approved before it hit a randomized trial. There would eventually be an RCT, but when it was sort of hitting the market, this would be based on evidence from an exclusively treated population. So this is great, but then what evidence do we have to show how M compares to existing therapies? So I want you to think about what we need to show cause and effect. Without a control group, can we just say, oh, the patients in our single arm trial, they had these outcomes? We can just read the literature and see what patients had when they took some other treatment. So maybe just think about my time machine example and think about why that's not really the best comparison. For example, what if the patients who took other medicines in the literature were very different from the patients in our trials? We don't have randomization to control the similarities between two comparison groups. And in our case, we also don't have a control group. So now I'll introduce the method that helps us move towards understanding the causal effect of this drug. Okay, so first things first, we need a control group. So let's create one. What we can do is use observational data sources like health registries and electronic health records to identify patients with the disease who are similar to patients in the trial. So one way we can do this is by trying to match the selection criteria of the clinical trial as closely as possible, such as if patients need to be a certain age or have certain disease characteristics. So we can basically source a population of what we'll call a real-world control arm. Now that we have a control group, the second problem is that we lack randomization. So our clinical trial patients may still have key differences from our real-world control patients. In other words, we would be comparing apples to oranges. For example, it's well known that patients who participate in clinical trials tend to be fairly healthy, and that real-world patients tend to be older, sicker, and have more heterogeneity in their disease characteristics. So this can bias our comparison of the two groups, and this bias is known as selection bias. So I'll just run through this new video. So first, I'll start by simulating a real-world control group of patients who are on average older and sicker than their clinical trial counterparts. So in this, I'll just sort of bias and sample older ages. I will not bias the sampling of sex, but I will also bias the distribution of ECOG score, which is a prognostic score for cancer, as well as the probability of having a heart condition. So just in here, I'm just going to show you that I've just simulated like 500 real-world patients, and I've added them to our clinical trial simulated treatment arm of 500 patients. Can I move next? I think my screen is paused. Okay, so what I wanted to lead you to was this plot here. So you can see pretty clearly now that patients in the non-randomized control group, which is the left panel, are more likely to have a heart condition. Look at the bottom left panel to see that kind of very skewed distribution. They're more likely to skew towards older ages in the top left, and more likely to skew towards higher or worse ECOG score in the bottom right. And then in my simulation, I didn't actually impose any difference in the distribution of males and females, so that's why the top right panel looks pretty even. So now we have our two groups that have not been randomized. One is the treated group from our single-arm clinical trial, and one is our control group of patients who are not in the trial. So these patients have different distributions of these different factors, which we believe could bias the treatment effect we want to estimate. Ideally, we'd like those factors to be balanced across groups so that we can make an unbiased comparison. So here, introducing a really elegant and simple method called the propensity score. So the propensity score measures the propensity of being in the treatment group, or the probability of being a patient in our clinical trial, based on the characteristics that maybe influence inclusion into the trial cohort. So it looks like this, the following formula: this probability of treatment given age, sex, other factors. And this model can take the form of a standard regression, in this case, a logistic regression, because the treatment is binary. It can also be a nonlinear model, a model with interactions, whatever functional form best captures this probability of treatment. The result is a conditional probability, so a number between 0 and 1, of resembling a trial patient, given everything you've adjusted for in your model. So recall that our trial patients are more likely to be younger and healthier than a real-world patient, because that's what I imposed in the simulation. When we develop this model, here's the profile of one of our simulated patients with a low propensity score. So see that they're age 59, they have a heart condition, high ECOG score, that's between 0 and 5, so 4 is pretty high. This patient doesn't look like a clinical trial patient according to how I simulated those patients, and so they were assigned a low propensity score. And you can contrast that with the profile of a patient with a high propensity score. They are age 23, no heart condition, ECOG score 0, which means good and healthy. They have a high probability of having been included in the trial or looking like a trial patient. So that's the propensity score. This is a busy slide, but really what the propensity score enables us to do is introduce comparability between our two groups through a kind of pseudo randomization. So we use the propensity score to derive weights for each patient based on how close or how far they are to a trial patient. So these weights are called inverse probability of treatment weights, or IPTW, because they involve taking the inverse propensity score. And so after weighting each patient, what this gives us is a reweighted patient population of patients who are similar to each other. So there are a few ways to do this, and here I show the formula for ATT, IPTW, which stands for average treatment effect on the treated. This equates to assigning basically a weight of 1 to the trial patients, that's our treatment group of interest, and reweighting only the control group. The result is that we upweight patients in the control group with a high propensity score, those patients who look more like trial patients, and we downweight patients who are very different from our trial patients. And the ultimate objective here is to create a control group that is as similar as possible to our trial patients in terms of those characteristics we adjusted for. So again, remember that our objective with causal inference is always to be able to isolate the effect of treatment, all else equal. Right, so here I've just pasted code instead of actually showing you a video, but from here it's pretty straightforward. So I'm still using our simulated patients and our 4 clinical characteristics. I use a logistic regression model to model the probability of being in a trial patient, and then derive IPTW weights using the equation that I showed in the previous slide. And just to illustrate that, I'll take those 2 patients I showed you on the other slide with low and high propensity scores and show the resulting weight in the final column. So you can see that the older, sicker patient was downweighted and the younger, healthier patient was upweighted. And when we apply reweighting across all patients, what we hope to see in the reweighted population are similar distributions of these baseline characteristics now. So I'll just kind of do this is pre-post. And so just maybe pay attention to the bottom left plot of heart condition, and you see how that goes from being very differently distributed to very similar pre-post. You also see kind of a more uniform age distribution, more uniform ECOG distribution. So this is achieving the reweighting that we really want to see. Once we've achieved that, we can now fairly compare outcomes between the 2 groups using any standard model applied to the reweighted population. So here I show examples for a binary treatment effect, like whether the patient showed a clinical response to treatment, or a time to event effect, like comparing survival between the 2 groups. So the interpretation of the average causal effect here is what hopefully brings this tutorial full circle. So say your outcome is response to treatment and you estimate an odds ratio of 2.5. You would say the odds of responding to treatment is 2.5 times higher for patients treated with M compared to had they been treated with standard of care, assuming some things which I'll get to in the next slides. But the key here is this would have been language. So that's our time machine. We made 2 groups as similar as possible so that we can say it's almost like we went back in time and treated the same group of patients with something else. And so that's how we isolate the causal effect of drug M in this example. But there are many, many limitations, which is why I said in the first slide that causal inference is both a bit of an art and a science. So first, when we say drug M has some causal effect on patients, we assume conditional exchangeability, which means we assume the 2 groups are similar given the characteristics we adjusted for. But what about, and in that case, it was 4 very simple variables, right? But what about the characteristics we didn't adjust for? So there's no guarantee that those characteristics are balanced, and therefore there can be residual bias in your analysis from unmeasured confounding. And so that's why proper randomization, like in an RCT, is really, the ideal scenario. It guarantees exchangeability on both measured and unmeasured factors. So here you have to spend a lot of time kind of thinking about and measuring the factors which you believe could confound your causal effect of interest. There are also a lot of statistical assumptions, some of them laid out on the right, like positivity and consistency. And then finally, we can only conclude for an average causal effect, sort of on average between two groups, because we don't have a time machine. We can't claim causality at the individual level. And despite the quite simplistic example that I showed here, it can get pretty complex. So I really like this excerpt from this great book written by Hernan and Robbins, who are considered the founders of causal inference. They kind of wrote, the more you learn and achieve in causal inference, the causal inferences you make become indeed less causal because of the many stipulations and assumptions that are rightfully couched around them. I just want to conclude with some great resources, which I will share actually on the Translational Science Slack channel, including what I think is the holy grail for causal inference, the book called Causal Inference, What If? So this is an amazing starting point. They also wrote out all of the exercises in R. I think they're also doing it in Python. So all around great resource. And so that is sort of concludes this talk. I look forward to the discussion, and Leah will actually follow me next and take us through possibly more complicated causal inference scenarios. So it should be a really good discussion.
Thank you.
Great. Sorry. Okay then. Hi everyone, and sorry, I just have to move something out of the way. Why can I not flick through? Okay, that should work now. Okay. Hi everyone, welcome to this presentation. Where I'll be talking about the issues of handling informative patient monitoring in routinely collected hospital data that's used to estimate longitudinal treatment effects. So I'll structure this talk as follows. I'll start by introducing the problem of informative monitoring in routinely collected hospital data. And I'll put this into context using a motivating example of invasive mechanical ventilation in COVID patients, describing the types of causal questions that we might want to answer using this sort of data. I'll describe the assumed data structure using a causal DAG, or a directed acyclic graph, introducing notation and assumptions. I'll then describe two potential causal inference methods that deal with informative monitoring in the context of estimating longitudinal treatment effects. And finally, I'll briefly share some results from a simulation study that I've conducted to compare these methods. So routinely collected hospital data provide the opportunity to study treatment effects that might not be feasible in randomized trials, for example, due to financial or ethical or logistical constraints, and that also may be more reflective of realistic clinical practice. And there's growing availability of these sorts of data. However, use of routinely collected data for estimating treatment effects requires causal inference methods to account for the complex confounding that occurs due to interrelations between time-dependent treatments and covariates. And also because the primary reason for collection of these sort of data is for ongoing clinical care rather than for research. Another specific challenge when it comes to routinely collected data is that measurements of patients' clinical status are made at high frequency and on differing schedules for each patient related to their clinical status. So the timing and frequency of monitoring is irregular and potentially informative about patient outcomes. So to use causal inference methods to estimate longitudinal or time-dependent treatment effects, we require longitudinal patient data where our treatment and confounders are measured repeatedly over the duration of follow-up. So ideally, we'd observe data at regular, evenly spaced observation times, like we see here for an example patient. So measurements of our confounder, LT, and our treatment status, AT, would be observed at regular intervals, for example, every 12 hours. But in practice, within routinely collected hospital data, typical measurements of treatment and confounders might look more like this. So this plot represents some possible observation times for an example patient, with red crosses representing measurements of a covariate LT and blue crosses representing measurements of treatment status AT. And these observation times can differ over time by covariate type and across patients. So a simple and crude way to deal with the irregular monitoring is to divide up the analysis time into a grid-like structure. So for example, 12-hour periods, as is shown here by the vertical dashed lines. And the length of the time interval will be chosen by the analyst. So where multiple measurements fall in a particular time interval, we might choose to take a summary measure, for example, the mean or median, depending on the type of variable. And also there will be periods where no measurements are observed. So the length of the time interval or the width on this grid will be chosen by the analyst. And so, for example, this plot shows what happens if we choose shorter 6-hour time intervals. And the result is fewer measurements being summarized in any one time period, but also more unmonitored time periods. And in the most extreme case, we could pick a very fine grid such that all the observation times fall on one of these vertical lines. So I'll now introduce the motivating example for this talk, which involves questions about optimum invasive mechanical ventilation strategies in COVID patients using routinely collected hospital data from patients in the intensive care unit. So we may wish to answer the following causal questions. Is invasive mechanical ventilation effective in reducing mortality in COVID-19 patients compared with no or non-invasive ventilation? And if so, when should patients be transferred to invasive ventilation? So this question of when to initiate invasive ventilation can be expressed in terms of different treatment strategies. So, for example, we may wish to examine the effect of initiating invasive ventilation within 24 hours of hospital admission, compared with maintaining patients on no or non-invasive ventilation only. And alternatively, we may wish to examine the effect of transferring patients to invasive ventilation when a value of some underlying variable, such as respiratory rate, 30 breaths per minute. And the first two strategies here are examples of static treatment strategies where patients receive the same pre-specified intervention. And the last is an example of a dynamic treatment strategy in which there are rules that adapt the treatment to the current status of the patient. So in this talk, I'll describe the methods in the context of static treatment strategies, but they can also be extended to dynamic treatment strategies. So in the next few slides, I'll describe the observed data structure under informative monitoring using a causal DAG or directed acyclic graph. And I'll introduce some notation and our causal estimand of interest and the identifying assumptions that we require in order to estimate this causal estimand from our observed data. So because the DAG is a bit complex, I'm going to sort of build it up in sections over the next few slides. So in this setting, we assume that we have this potentially very fine grid, like we saw before, where the measurements, where collected, always fall upon this grid. And this could be, say, minutes, hours, days, etc. in our data. So here we have like a simplified setting with just 3 time points. And we have some time-dependent binary treatment, A subscript T, time-dependent covariate L subscript T, and then a final outcome Y. And we can think of LT as representing the observed covariate. So LT isn't necessarily monitored at every time point for all subjects. And then I'll introduce here a new variable, N subscript T, which is a binary variable representing the decision to monitor LT at the next time point. And as well as the observed covariate L, we also assume that there's a true and underlying covariate L star, which is sometimes unobserved. So at the first time point, we assume that L0 is always monitored, for example, because this may form part of our eligibility criteria. So we have L0 equals L0 star. And it's this underlying true covariate process that affects Y. So we have arrows going from L star to Y. And also because the treatment may possibly affect the outcome, we have arrows from A to Y. And similarly, A affects the true underlying covariate L star. However, we assume that the covariate can only affect treatment decisions when it's observed. So there are no direct arrows from L star to A, only from L to A. And this is because any treating clinicians or doctors can only make decisions about patients' treatment based on variables that have been monitored and therefore observed in the data. And similarly, N can be affected by the observed covariate L, as well as the treatment and past monitoring decisions. And in turn, N can also impact future monitoring and treatment decisions. And then finally, the dashed arrows represent deterministic relationships. So for example, L1 is equal to L1 star if N0 is equal to 1. So if the decision was made to monitor the patient at the next time point. And otherwise, L1 is equal to L0 because any treatment decisions at time 1 will be made based on the last observed value of L. So the causal estimand of interest can be expressed as some contrast of the expected outcome under different hypothetical treatment interventions. And we call these potential outcomes, which are the outcomes that would have occurred if the treatment had been assigned, possibly contrary to fact, to some treatment intervention of interest. So we use superscripts here to denote these potential outcomes. For example, here we're going to look at the causal contrast under the treatment strategy always treat, i.e. where A is set to 1 for all times, versus the treatment strategy never treat, i.e. where A is set to 0 for all times. And the following assumptions are necessary to identify this causal estimand from the observed data. So note here that we use an overbar to denote the history. So here A bar is just equal to A0, A1, and A2 combined. So the first assumption is consistency, which says that for a patient who actually received the treatment strategy that we're interested in, the patient's observed outcome will be the same as their potential outcome. And in practice, this normally means that we have a clear and well-defined treatment intervention. Then we have conditional exchangeability, which states that the potential outcome is independent of the actual treatment assignment at time t, conditional on all past observed covariates, past monitoring decisions, and treatment history. So this means in practice that we have no unmeasured variables that affect both the treatment and the outcome. Then positivity, which states that given a patient with any combination of observed covariate monitoring and treatment history, there's a non-zero probability of that patient being both treated or untreated at the current time. And finally, the assumption of no direct effect, which states that the decision to monitor a patient won't affect the underlying value of the covariate being monitored, i.e. L star, or the outcome. So we don't have any direct arrows from N to L star or from N to Y. So I'll now introduce two causal inference methods that we can use to estimate this causal estimand from our routinely collected hospital data, which is subject to informative monitoring. So the first method I'll describe is inverse probability of treatment weighting. And with inverse probability weighting, the basic idea is to break the link between the confounders and the treatment, i.e. the orange arrows in this DAG, by reweighting subjects in the analysis to mimic a situation in which the treatment is assigned at random. And we do this by calculating time-dependent weights, which are the inverse of the probability of being treated, conditional on past treatment, monitoring, on observed covariate history. And here we exploit the fact that treatment depends on observed covariate history and not on the underlying covariate L star. So the models here include L rather than L star. And the steps involved in calculating the causal estimand of interest are, firstly, to fit models for the observed data. So for A, conditional on L0, and then for A1, conditional on past A, N, and L, and then for A2, conditional on any past A, N, and L. Then for each subject, we calculate the predicted probabilities based on their actual treatment assignment at each time t, i.e. the probability that A equals 1 in subjects who actually received the treatment at that time point, and the probability that A equals 0 in subjects who did not receive the treatment at that time point. We multiply these together and take the inverse to estimate the weights for each subject. And then we reweight the subjects by multiplying their outcome y by their estimated inverse probability of treatment weight. And then we average over these reweighted y's within the individuals who followed the treatment strategy that we're interested in. So for example, here we're considering the always treat strategy. The second method I'll talk about is G computation. So G computation involves breaking the link between the confounders and the outcome, i.e. these pink arrows here, by sequentially modeling the confounders at each time point and then simulating data under the hypothetical treatment intervention of interest. So based on the identification assumptions of consistency, conditional exchangeability, and no direct effect, it can be shown that the quantity we want to estimate, i.e. the mean of the potential outcome, can be expressed in terms of things that we can estimate from the observed data. So although this formula looks a bit complicated, basically we have, because y depends on the true underlying covariate L star, we can see that we need models for y given the past treatment and confounder L star, and models for the confounders L star over time, given things that we observed earlier in the DAG. And to implement this method in practice, we start by imputing missing values for the unobserved L star sequentially in time, first for L1 star and then for L2 star using the imputed values for L1 star. We then simulate values for L1 star using the fitted model, but with A0 set to 1. So we're simulating what L1 star would have been if we'd intervened on A0. And then similarly, we simulate values for L2 star from the fitted model, but with A0 and A1 set to 1, and with L1 star set to the simulated values. And finally, we take the fitted model for Y, given the observed data, and obtain fitted values under the treatment intervention of interest. So with A0, A1, and A2 set to 1, and with L1 star and L2 star set to their simulated values. And this gives us the potential outcomes for Y, and we can simply average over all individuals. And we can use bootstrapping to allow for uncertainty and imputation in imputing the unobserved L star. Alternatively, we can implement this G computation method without requiring imputation of L star by exploiting the fact that L star is only a confounder when it's observed. So here we stratify the data by each monitoring pattern, and then we fit models for our observed covariate L1 and L2, and for the outcome Y, conditional on any past observed covariate monitoring and treatment history. And then we can repeat the G computation steps just described in the previous slide, but this time within each monitoring pattern to simulate Y under the hypothetical treatment intervention of interest. And we can combine results across the monitoring patterns to get our estimate of our expected potential outcome. So in the last few minutes of this talk, I'll very briefly describe a simulation study that I conducted to compare these methods and present some of the results from the study. So in terms of design, the estimand of interest was the difference in expected outcome under the always treat and never treat strategies. And I generated data sequentially in time according to the data structure set out in the DAG described previously for both 3 time points, which I demonstrate here, and then also for 12 time points. And I considered a single continuous covariate L star and a continuous outcome Y measured at the end of follow-up. So true values of the average treatment effect were obtained using a simulation approach, i.e. by simulating the data in a similar way to the DAG, but instead by setting AT by intervention to the treatment strategies that we're interested in. And then I applied the two methods just described, as well as some naive versions of these methods where the monitoring variable is completely ignored. So the plot shows the mean average treatment effect across all 1,000 simulations for each of the methods compared, and the corresponding empirical standard error and 95% confidence intervals. And the blue dashed line shows the true average treatment effect. So the methods that I described in this talk, which are the IPW method in orange and the two ways of doing G computation in pink, reduce bias compared with methods that ignore informative monitoring, which are shown by the grey dashed lines. And the confidence intervals based on the empirical standard errors, which are the standard deviations of the estimates across all simulations, are wider for IPW-based methods than for G computation-based methods. Thanks very much for listening. Yeah.